questions to ask. So with the logistics out of the way, please allow me to introduce to you our Howard S. Brody lecturer for this afternoon, Dr. Monica Uddin. Dr. Uddin joins us from the University of South Florida, where she is a professor in the Genomics Program at the College of Public Health. Professor Uddin earned her BA in human biology at Stanford University and her PhD in molecular anthropology at New York University. Professor Uddin's remarkable research focuses on how traumatic stress and social adversity can affect the regulation of human genes in ways that can stretch across generations. In her talk today, Professor Uddin links traumatic experiences in African and African American populations with changes in epigenetic processes that can lead to an increased risk for mental illness in subsequent generations. Her remarkable work deploying historical, anthropological, and molecular biological techniques has far-reaching implications. It forces us to rethink the timeline of social impact and how we approach humanitarian crises. From my perspective as a neuroscientist, I can tell you that Dr. Uddin's work challenges us to consider the long term, and by that I mean across generations, beyond a single generation. To consider the long-term consequences of trauma and disorders like post-traumatic stress disorder. Her work makes clear the need to consider mental health from historical and societal perspectives and to rethink the imperative for mental well-being as we imagine future societies. I can go on about how amazing Dr. Uddin's research is, but you don't have to take my word for it. Professor Uddin has authored nearly 100 peer-reviewed publications and has secured over $10 million in grant funds to support her research program. So her work is impactful. And without further ado, it's my sincere pleasure to introduce Dr. Monica Uddin, who will be speaking today on trauma, adversity, and the epigenome, looking within and between generations into African ancestry populations. Monica, to you.
Thank you so much, Lena. Can you hear me? I'll take it that you can hear me. I appreciate it.
Yes, we can.
Okay, thank you. And I will do my best to live up to the very generous introduction that you gave me. I'm very pleased to be here speaking with you in this very important seminar series on race, violence, and health. And I'm excited to be a part of this really important work that you all are doing at Whitman College. So as Lena said, my work involves broadly understanding the biologic embedding of stressful and traumatic experiences. And today I'm going to focus on two specific examples of that, one in a US population of African Americans located in Detroit, and another in a Rwandan population in Africa. This is the outline of what I will be presenting. I want to spend a decent amount of time on background so we can all have the same vocabulary moving forward when I get to some of the data-based information that I'm going to share with you today. So I'll talk about post-traumatic stress disorder, or PTSD, and trauma exposure, why it is we might want to study epigenetics in relation to these factors in the first place. And then I'll move into the specific examples that I mentioned. The first being examples of stress, trauma, and social adversity in the Detroit Neighborhood Health Study. And then the second being the intergenerational impact of exposure to the Rwandan Genocide against ethnic Tutsi. Finally, on a more positive note, I will talk about genomic diversity and the contributions that the H3 Africa Consortium is making towards increasing efforts towards genomic diversity, and share with you some concluding thoughts. I'll move fairly quickly through some of the slides, and I won't have time to go into a lot of details about certain of the methods, but there are, in some instances, preprints of the work that I'll be discussing on bioRxiv, should you be interested in learning more about it on your own time, and I'm happy to answer any questions as well. Okay, so PTSD. So I think by now, most people have a sense of post-traumatic stress disorder just virtually by reading the newspaper every day. PTSD is defined psychiatrically as a combination of symptoms that occur after exposure to a traumatic event. Now, I want to pause here and just say that a traumatic event is something qualitatively different from a stressful event. A traumatic event is one in which you experience life threat or you witness somebody else's life being threatened. It can also include sexual and violence exposure. So it is different, for example, than losing your job suddenly. Although that is certainly a stressful event, it wouldn't be considered a traumatic event. There's a combination of symptoms that occurs after a traumatic event. And if you endorse enough of these symptoms, you would qualify for a PTSD diagnosis. These include symptoms of re-experiencing, avoidance, negative alterations in cognition and mood, and alterations in arousal that weren't there prior to exposure to the trauma, that occurred following the trauma, and that persisted for at least 1 month. So the interesting thing for me as a scientist is the fact that a majority of adults are exposed to a potentially traumatic event during their lifetimes, but only a minority of us develop PTSD. And so there's clearly factors that distinguish people who are vulnerable versus resilient to trauma in terms of their likelihood of getting the disorder. And understanding that from a scientific perspective is particularly interesting. So as mentioned, I work in a college of public health, so it's very important for me to contextualize disorders such as this in terms of the public health impact. If we consider lifetime PTSD prevalence globally, there's about a 13% prevalence. Within the US, that figure is closer to 7%. There's a high economic costs in terms of disability adjusted life years. So that those are the loss of years due to premature death, disability, or other ill health that wouldn't have occurred had people not had PTSD. So PTSD isn't as impactful as something like depression, but it still does indeed make an impact towards dailies as calculated by the World Health Organization. And as we know from a decent amount of work now, there are significant medical comorbidities that go along with PTSD. Some of these are other mental disorders, such as major depression, and other factors are things like cardiovascular disease or diabetes, things that at first glance may not seem to go along with PTSD, but we know from prospective studies actually can be brought on by having the disorder. So what about trauma? I mentioned that trauma is quite common. About 70% of us around the globe by the time we're adults have been exposed to at least one trauma, but there is variability in that figure. And so you can see here, for example, Northern Ireland reports about 60.6% exposure to trauma among adults in that country. USA is higher at about 83%. And what does that mean in terms of PTSD? It doesn't always translate that the countries with higher trauma exposure have higher rates of PTSD. Northern Ireland here, as you can see, has a higher 12-month prevalence of PTSD than does the United States. So there's, again, this disjunct between exposure to trauma and prevalence of PTSD that's really interesting to understand. Another thing to consider is the variability of types of traumatic event exposure. And what I'm showing here is a recent population-based study that looked at race ethnic differences in exposure to trauma types within the United States. And so it's a population-based sample, which means that it's representative of the US population of adults. And what's interesting is that on the whole, African Americans are actually less likely to be exposed to any trauma when compared to whites. But when you break it down by specific types of trauma, here I have indicated with a star which ones these are: participation in organized violence, sexual violence, and other. You see that there are significant Black-white differences with African Americans being exposed to higher rates of those types of events. Now, what does that mean in terms of lifetime PTSD prevalence? So this is another population-based study that's confirming the same thing as the earlier slide I showed you, but in a different way. This is the prevalence of lifetime PTSD by race ethnicity, and this is an even bigger population-based sample of over 34,000 individuals in the US. And what you can see here is this black bar indicates prevalence of PTSD among African Americans. It's about 8.7%, and that's significantly higher than whites, as is the bar showing here, which is for Asians. It's significantly lower than whites. Now, it's easy to potentially interpret this higher prevalence of PTSD due to African Americans' higher exposure to interpersonal violence, the slide that I showed you previous to this slide. IPV has a higher conditional risk of PTSD, meaning that if you're exposed to those types of events, as opposed to maybe getting in a car accident, the likelihood of you getting PTSD is higher. But what this study shows in data that I'm not presenting and the prior study as well, is that even when you control for those trauma characteristics, such as type of trauma, severity of trauma, age at first trauma, this higher conditional risk of PTSD among African Americans remains. And so there is something going on that needs to be examined more in detail to understand what that is. There could be mechanisms that have been hypothesized, such as exposure to social adversities, discrimination, feelings of isolation, social support, but there could also be biological factors that we don't yet understand that work in concert potentially with social adversity to shape higher risk of PTSD among African Americans. So what do we know about what causes PTSD? This is a far more general slide that synopsizes twin studies that have examined the genetic and the environmental contributions to this disorder. And you can immediately see that the environment has a far greater contribution to risk of this disorder than does genes. Now, it doesn't specify which environmental factors or which genes shape that risk, just that both are important. Now, as we know, or as I will explain if you may not be aware, epigenetics is actually a biological mechanism that can mediate environmental exposures for complex disorders such as PTSD. So in this slide, what's captured by the environment may potentially include epigenetic factors as well. Okay, so why epigenetics? Like PTSD, you may have heard a lot about epigenetic mechanisms recently in the news. It's getting a lot of press because whereas maybe 20 years ago, we had a lot of faith in understanding how genetic variation shaped risk for disorders, we were sort of disappointed that there wasn't a higher impact of many of the findings that were genetically based. Now, as we're learning about epigenetics, where we once thought that epigenetic factors were set early in life and did not change, we now understand that these molecular tags are modifiable and responsive to the environment. So these are mechanisms that alter or regulate gene expression without actually modifying the underlying DNA sequence. They can regulate DNA accessibility, and as I mentioned, they're influenced by age, environmental exposures, and disease. And what I'm showing here on this slide is a few different types of epigenetic mechanisms, histone modifications at the top, non-coding RNAs, specifically microRNAs in the middle, and then DNA methylation down at the bottom. But the key thing to remember is that they do nothing to the underlying DNA sequence, but they do control how genes get turned on and off. Okay, so among those mechanisms, what I'll be talking about today is DNA methylation. So DNA methylation is the most stable of the three mechanisms I just mentioned, and it refers to the addition of a methyl group to the 5' position of cytosine when cytosine and guanine co-occur on the same DNA strand. So that's an example here. So this is one strand of DNA paired with another, adenine and thymine, cytosine and guanine. When cytosine and guanine occur next to each other, that's called a CpG site, and it enables methylation to occur on the cytosine. It can occur in non-CpG contexts as well, but it is most commonly found at CpG sites, and that's what I'll be talking about today. Like the other epigenetic mechanisms, it's known to alter gene expression, and it is tissue specific. So this is something to remember as we go on, because what I'll be talking about are DNA methylation measurements in the blood, which is a peripheral tissue, but oftentimes we want to know what's going on in more distal tissues that are harder to reach, such as the brain for something like PTSD. So when we make inferences based on what's going on in the blood, we have to be careful not to assume that they reflect what's going on in the brain, because it is tissue specific. Okay, a little bit more about why epigenetics. Well, unlike most mental disorders, PTSD actually requires an environmental exposure. If you have not endorsed exposure to trauma, you are not eligible for a PTSD diagnosis. That's very different than the vast majority of other psychiatric disorders. It's true that exposure to trauma will increase risk for depression, but it's not a requirement of the diagnosis. So PTSD, by definition, requires an environmental exposure. And epigenetic mechanisms are responsive to the environment and may help to explain observed differences in risk for PTSD. We know from prior work that epigenetic differences in blood seem to fall into biological processes that are previously implicated through other biological work in PTSD, for example, in genes related to immune function and response to stress. And EWAS studies, which are epigenome-wide association studies, which take snapshots of DNA methylation profiles across the genome, can help us to identify biological pathways that are actively being regulated in PTSD and can potentially be targeted for treatment. And so it's another layer of genomic information that can be studied separately from genes or in concert with genes to understand how risk for something like PTSD is shaped. Okay, so now on to a specific example of how this might be important to a study of stress, trauma, and social adversity in the Detroit Neighborhood Health Study. So the DNHS, as we like to call it, is a study that's been ongoing for some time. It was launched in 2007 as a population-based study of adult residents of Detroit, wherein just over 1,500 adult participants were recruited and followed for 4 subsequent years. The baseline wave was comprised of a 40-minute structured telephone interview, and it was really wide-ranging. It included questions about people's exposure to trauma, PTSD symptoms, depression symptoms, anxiety symptoms, but also questions about their social support, questions about their perceptions of their neighborhood, questions about other aspects of their health, physical health, questions about their drug use. So the idea being that we wanted to capture a really rich, detailed snapshot of what was going on in the population of Detroit at this time that went beyond potentially a typical psychiatric epidemiology study. So I won't be talking about this today, but survey participants were geocoded and linked to their neighborhoods based on information on their residence location. And there's been a lot of work in this cohort done at the level of the neighborhood, which is unique among cohort studies. And then a subsample of participants provided a biologic specimen at each wave. So people who consented to be visited in their home had either a venipuncture blood draw or a blood spot or a saliva sample collected at 3 of the 5 waves. So we weren't, unfortunately, funded to do bio collection during all the waves, but we did what we could, and we have multiple repeated measurements for many participants. So this is a snapshot of each of the participants in our baseline wave at the city of Detroit. And it's just to show you that we had quite broad representation across the city. The blue in the middle is actually Hamtramck, its own city incorporated smack in the middle of Detroit. And this yellow island here is Belle Isle, and it's non-residential. So that's why there are no red dots there. So that was the original study that was funded by NIDA, National Institute of Drug Abuse. A few years ago, we were lucky enough to seek and secure funding from the National Institute of Minority Health and Health Disparities, looking to examine some of the biological specimens we had collected but weren't funded to actually analyze with this specific research question that you see in front of you here. So we wanted basically to understand better how the interaction of social adversity and epigenomics may impact subsequent risk for psychopathology. And so we focused in on a specific pathway of glucocorticoid receptor regulatory network genes, which I'll describe more in a minute, as a potential mechanism linking these 2 factors, so social adversity and stress-related psychopathology. And we did this by drawing on extant specimens we had collected in the parent study, and we collected new epigenomic data with one of the newer technologies that had emerged and paired it with the survey data that already existed from the parent study. And why did we focus on the GRRN pathway? So we focused on that pathway because we wanted to look at stress-relevant DNA methylation that would have relevance in blood. And we know that glucocorticoids are a hormone that are produced by the adrenal gland and distributed in the blood throughout the body. And this is the output of the HPA axis. So when you're stressed, the hypothalamus will produce corticotropin releasing hormone, send it to the pituitary, which will produce adrenocorticotropin hormone, which sends it to the adrenal gland. And then that produces glucocorticoids, chiefly cortisol, which is something you've probably all heard about. And that then gets dispersed in the blood to regulate gene expression in almost every cell in the body. And so there are 82 genes that have been mapped to a regulatory network that kind of controls this glucocorticoid receptor, and they have functional relevance in the blood. So yes, we're interested in the brain because we're studying PTSD, but we're looking at living individuals. We cannot access their brain. So we look at biological pathways of high relevance in blood because the tissue is relevant to our question. So we also capitalized on this pathway because a lot of prior work had implicated it in PTSD as well as other stress-related mental disorders. So we use that prior knowledge to focus in our pathway of interest. And this was our working model. What we wanted to do was examine the joint effects, if you will, of factors that had been previously implicated in increased risk for psychopathology, meaning socioeconomic position, stressful life events, psychosocial stressors, and then see how they may work in concert with DNA methylation in the GRRN pathway to elevate stress-related mental disorders. For the example I'm going to be talking about today, we're focusing specifically on post-traumatic stress symptom severity. So the research question of relevance for this study, there are two. So what are the significant factors among social adversity, psychopathology, and GRRN DNA methylation that are associated with prospective risk of PTSD? And two, how well can we predict the prospective risk of PTSD using the factors that we identified as significant? And this work was led by a postdoc in my lab, Agazwani, and the title of the paper that you see there is under review, but there is also a preprint that's been posted to bioRxiv if you're interested. So this is just a quick snapshot of the DNA methylation data. I don't have time to go into the details, but this is an array-based platform where you can collect data on 850,000 CpG sites at once, and DNA methylation is measured as a beta value, which can be measured from 0, which is completely unmethylated, to 1, which is completely methylated. If something measures as a 1, it means that methylation is basically 100% at that site. And if that occurs in something called a promoter region, it likely silences the gene. Conversely, if the beta value is close to 0, it means if it occurs in a promoter region that it likely enables the gene to be turned on. So for the analysis I'll be describing in just a minute, we use DNA methylation data from waves 1 and 2, and we use social adversity and psychopathology data from waves 1, 2, and 3 to predict post-traumatic symptom severity at wave 4. And this is the sample that we used. We had 148 unique participants. 62 participants had 2 time points of DNA methylation data. So not all our participants, but a good number of them. The sample was 60% female. Average age was about 55. And the overwhelming majority of the participants in the sample was African American. And you can see too that the cumulative number of traumas is quite high, almost 7. Now, usually PTSD is endorsed in relation to a single trauma, so your symptoms will be reported in relation to that trauma. But we also know that the more trauma you experience, the more likely you are to get PTSD. So this is a relatively highly traumatized sample that we're looking at. So how did we examine how these factors worked in concert with each other? We used machine learning approaches, which again, you may have been hearing about more these days. These are approaches that can have supervised learning or unsupervised learning or reinforcement learning approaches to understanding the data. And the advantage of machine learning is that the approaches learn from the data itself. It doesn't require explicit programming to use if-then statements if certain conditions are met in the data. It learns from the data to train itself to create a model that best predicts what your outcome is. And for our purposes, we used supervised learning because our data that we provided as input had labels, and our data that we had for our outcome also had a label. Okay, so we had two types of different data sets, and I think it's going to be too complicated to get into what the differences are. It was just a different way of structuring the matrix as either wide or long if somebody had more than one methylation measure. But in either case, you can see that the features we were using were quite numerous, over 2,700 at least in the smaller version of the data set. And that's a lot of features to put into a machine learning model and expect it to run efficiently. So a key pre-processing step in machine learning is to identify important features. And we did this by conducting univariate tests for each feature's association with PTSS. And we retained the top 150 features in each of our data matrices. And they weren't the same set in each of the two matrices, but there was a decent amount of overlap. So in the long data set, we had 79 CPGs and 71 phenotypes. In the wide data set, we actually had more CPGs chosen as important and slightly fewer phenotypes, but 95 in common between the long and the wide. And then we used these features to predict, using machine learning approaches, post-traumatic symptom severity. And this is a huge slide that I don't expect you to be able to see very well. I just wanted to show you a brief overview of the 95 significant features, both CPGs and phenotypes, that showed up to be common in both long and wide analyses. In general, the importance of phenotypes were greater than the importance of individual CPG sites, but there were exceptions to this pattern. Okay, so what I'm showing here is a zoomed in version of that previous slide. So I'm showing you the top 10 significant features common in both long and wide analyses. And these are shown in negative log scale. So closer to 0 is more important, which means that the factors listed up at the top of the graph are more important than the ones listed at the bottom, but they are all important. And in the gray bar, we're showing you the importance in the long data set, and in the white bar, the importance in the wide data set. So you can immediately see that post-traumatic stress symptom severity in wave 3, the wave immediately prior to wave 4, was the most important predictive factor in predicting our outcome. There were other genes, other psychopathology factors that also figured prominently, wave 2 PTSS, wave 1 hyperarousal, that were shared in both analyses, and then CPG sites that fell into distinct GRN genes, namely AKT1 and FKBP5. So FKBP5 has been very heavily implicated in a range of stress-related mental disorders, including PTSD. So that was the top 10 features common to both long and wide analyses. What I'm showing here are the top 10 features that are in the long and wide analyses that aren't necessarily shared. But I want to draw your attention to the top row because it echoes the previous slide. Wave 3 PTS symptom severity is most important, but this number here shows the degree of importance. It accounted for 89% of the prediction of PTSS. So in the many features that were identified as important, 150, which weren't necessarily the same in the long and the wide analyses, the total amount is going to scale to 100. So that means that this feature has an extremely heavy weight in predicting PTSS, 89% of it. And then everything else is important, but far less important because there's going to be 149 other features that have to add up to basically 11%. But you can see there are some differences. So the first two features are still the same as in the last slide, but then here we have wave 1 intrusive symptoms as being important, and on the right we have hyperarousal. But by and large, the most important factors are prior PTSS. I will say, and I can't show you because it's hard to visualize, that there were social adversity factors that were also important. So the top ranked social adversity feature was loneliness. It was ranked 28th out of 150. in the shared set of important features. Perceived discrimination was 43rd, and financial problems was 63rd. So all of these features, although they're not shown here, were at least in the top 100 that were selected. Our work also did a lot of fine-grained evaluation of the different machine learning models. We tried 7 different ML models that were tested in both the long and the wide data sets. The best performing model was a gradient boost model. It had R-squared of 95%. And I encourage you to look up the preprint if you're interested in those aspects of the work. So this work is based off of what we call batch 1 of our data. We have another batch coming in later this month, and we will be able to do additional machine learning work on an even denser data set than what we presented here today. But based on what we have seen, we know that machine learning models can incorporate a really rich range of psychopathology, social adversity, and genomic factors that you can look at in concert to predict prospective risk of something like PTSD. It's very clear that prior post-traumatic psychopathology has the greatest weight in predicting future risk, which is consistent with previous work. But there were some social adversity factors that were also implicated. So once we get the second batch of data, we'll be able to augment this work and maybe even have additional features that are identified with finer grained data. Okay. Now I'd like to move on to the next example, intergenerational impact of exposure to the Rwandan genocide against ethnic Tutsi. So I realize that many of you listening may not have been born when the Rwandan genocide against the ethnic Tutsi occurred. So I want to just give you a brief bit of background. So this was approximately 100 days of killing that occurred between April 7th to mid-July in 1994. And about 800,000 people were killed, 1/7 of the population. Some think that that's an underestimate. And these were largely ethnic Tutsis that were targeted by Hutu extremists, but Hutu sympathizers were also included in that figure. Nearly 2 million people were accused of participating. It involved heavy involvement by the government at the time. And the result is that 1 in 4 children were orphans based on data from the census in 2005. And 26% of the population had PTSD, with the figure being even higher among orphans and widows. So obviously this is a hugely impactful historic event, and the repercussions of this continue to be felt. Many of you may have heard about the genocide in Rwanda and may only associate that country with that really dark time. But I encourage you, if you ever get an opportunity to visit, please do so. The country is amazing. It's really one of the economic engines now of Central Africa, and is a place where people from all over the world now are coming to start their businesses. So this is an important bit of history, but it shouldn't be obscured by the progress they've been able to make despite this dark period. So this episode in their history created the foundation for one of my collaborators to reach out to me and ask if I would partner with him to investigate the intergenerational impact of the genocide in women who are survivors of the Rwandan genocide and their offspring. So this is a large consortium effort that is being funded by the National Institutes of Health and the Wellcome Trust. That's the H3 Africa consortium that I'll talk about more in a minute. This particular study, which is a smaller study, is actually funded by the National Institute for Mental Health and the National Human Genome Research Institute. And the goal is to, 1, build scientific capacity. That's a key feature of all H3 Africa awards. And so that's one of the drivers of our award too. And 2, to conduct a modest study on the intergenerational genomic impact of the genocide. So the overall project design involves training and infrastructure, and that's the scientific capacity building that I mentioned earlier, community and public engagement, so interaction a lot with our funding agencies. We have consortium meetings twice a year. we engage with study participants, not me personally, because I need to be separated from that and only deal with de-identified data, but people on the ground in Rwanda engage with the community to let them know about the study and help them understand the impact of the genocide on mental health. And then the research component is, of course, what's still ongoing, a little bit delayed because of COVID, where we recruit participants, collect samples, and conduct psychological analyses. So this is our study team. It doesn't even capture everybody involved in the study, but this is just to show you that it really takes a village. And the focus here is on understanding the impact of early exposure, and by that I mean prenatal exposure to the genocide, on long-term health. So what I'm showing here is actually in relation to type 2 diabetes, but it was the best depiction I could find of how epigenetic factors could influence possible complex phenotypes later in life. So if you think about it, during pregnancy, right after conception, you're a zygote, you're one cell. You have to go from being one cell to a newborn in 9 months. That takes many, many, many rounds of replication of DNA. If during pregnancy, a mother is exposed to unbelievable trauma like a genocide, and if that exposure alters the in utero milieu in such a way as to impact a DNA methylation level of a particular gene, that occurrence can get propagated through multiple DNA replication rounds such that when a baby is born, it will come into this world potentially with the DNA methylation levels set in response to that exposure. And this can involve things like DNA methylation. It can also involve other epigenetic mechanisms as well, in that setting in relation to the in utero exposure can then potentially predispose that individual to later risk or resilience to mental disorders. And my collaborator and study PI, Leon Mutesa, conducted a pilot study testing just this idea. So this is a paper that was published in 2014, where he looked at a sample of 25 mothers exposed while they were pregnant to the Rwandan Genocide and their offspring, and then a matched control group of ethnically Tutsi women who were out of the country during the genocide, pregnant during the genocide, and their offspring as well. And this is just showing you that mothers who were exposed to the genocide had elevated rates of PTSD and depression compared to mothers not exposed. Children had the same pattern. He did a focused, locus-specific analysis of DNA methylation in the glucocorticoid receptor. So that's a key stress-relevant gene that is the one that binds to cortisol and helps tamp down the stress response. And what you can see here is that there are discrete CPG sites, the ones that have these little symbols, that showed significant differences in certain comparisons. So the hashes indicate highly significant CPG sites showing differences between exposed children and non-exposed children. And then these little stars are the exposed mothers compared to non-exposed mothers. So not all the sites had differences, but there were clear instances where CPG sites in this hugely stress-important gene showed DNA methylation differences. When he looked at plasma levels of cortisol, the exposed children and mothers had reduced cortisol levels, and that's a hallmark of PTSD pathophysiology. In our current study, we will be looking at a different kind of study design. We will have mothers who were pregnant during the genocide and in Rwanda, and the child born from this pregnancy. Will also be recruiting women who have a younger child after the child who was in utero during the genocide, so that we can potentially test for persistent effects that existed beyond the in utero exposure. We'll have a control group, and I should say that this study is collecting these samples from consenting participants as part of the biobank effort going on in H3 Africa. So all these samples are being collected and given to the biobank so that specifically African PIs can have access to it exclusively for 3 years. Our study actually doesn't have funds to test all these samples, but we're collecting them for the benefit of the larger African scientific community. So this is a picture from our first community engagement event. These are some of the women who came, and the purpose was to just inform them of the study and what the goals were. They weren't necessarily study participants, but certainly many of them had lived through the genocide. So the research objectives for our methylation study, which were really only just beginning, you can see here. So this is sort of uncharted territory for human studies. Most intergenerational studies have focused on animal work for obvious reasons. The generations are much smaller, you can control things. So what we were interested in looking at is just determining the genome scale methylation levels that associated with exposure versus non-exposure to the genocide using this epigenome-wide approach. And then possibly to identify epigenetic differences associated with intergenerational trauma. So the pilot sample that Dr. Mutesa already reported in 2014 actually had DNA still from many of the participants. So the first step that we're doing is to look at the DNA from those participants to see if we can identify these genome scale patterns. So we had 70 participants with adequate DNA, 31 who would count as exposed, and 39 who would count as non-exposed. And again, we use the same array technology as in the previous example. And this is the pipeline. So this work is being led by Dr. Agazwani and PhD student Clarice Musana-Baganwa, who's a trainee on the grant. It's a really small sample to start with for a EWAS analysis, and only 59 samples survived QC. So we wanted to focus in on the top 5% of probes showing the greatest variance, because when you're testing for differential methylation, if a CpG site has the same methylation level in everybody, you're not going to identify any differences. So if you hone in on those that show variation in the first place, you may be more likely to identify differential methylation. We focused on differential methylated regions, DMRs, as opposed to discrete CpG sites. And we identified 16 significant DMRs in the children that were associated with genocide exposure and 24 in the mothers. And then we did a little bit of independent tests of individual CpGs just to see what was going on. And this is definitely preliminary. We need to follow up with more samples and more analyses, but I'm sharing it with you today as sort of a preview of where we're going. So this is a fairly boring table just to show you the 24 genes that were identified in association with the DMR analysis in mothers. We had 16 in children, and we had 6 that overlapped between the mother and the child analysis. So these 6 genes here were identified as differentially methylated regions, both in children exposed versus unexposed to the genocide and mothers exposed versus unexposed. And what I'm showing in red is the fact that they had the same direction of effect in both the children and the mothers, which is interesting. Now, of course, again, this is all in blood and we're interested in brain. And now there are ways to correlate what's going on in the blood with what's going on in the brain using databases that didn't exist 5 years ago. And so of those 6 genes that I just showed you, 3 of them actually show strong correlation in DNA methylation levels between blood and brain. The first one, PM20D1, has been implicated in Alzheimer's disease and Parkinson's disease. And this is actually the only one of the 3 that survived multiple hypothesis test correction for all the CpGs that are correlated between brain and blood. So this one would be what we would likely follow up the most if it's borne out in additional work. And this is just pulling out a single CpG within the region that was implicated as differentially methylated to show you the pattern in the children and the mothers, where those who were not exposed to the genocide had lower methylation levels than those who were exposed. Now, there's a fundamental problem with everything I just showed you, and that is we haven't yet taken into account the relatedness between mothers and offspring. So our next step is to conduct some linear mixed models controlling for family as a random effect to see if what we see in the mothers is predictive of the children. But this is just a first pass. So to summarize here, based on Dr. Mutesa's earlier work and the literature in general, we know that abnormal HPA access activity really plays a central role in PTSD pathophysiology. It is possible that epigenetic modifications underlying these dysfunctions contribute to the transmission of traumatic stress from mother to offspring. In our current pilot analysis, we identified many common differentially methylated regions in both children and mothers that were associated with genocide exposure. And what we know just from Dr. Mutesa's earlier work alone is that there is a biologic embedding of exposure to the Rwandan genocide that is detectable and endures more than 20 years after that terrible event. So his paper was published in 2014. He collected those samples in 2011, 2000 to 2013, so about 20 years. So these are long-lasting effects that we're seeing, and again, they're in blood, so we can't really know for sure what's going on in the brain, but if nothing else, they are biomarkers of exposure to the genocide in this particular population. So I know we're running out of time, and I'll just try and go really quickly through the last bit. So genomic diversity. So I work in public health, and public health is definitely interested in understanding how diversity impacts health outcomes. And there's been growing interest in genetics, obviously, as one metric of diversity, but we are suffering from a lack of diversity in our publicly available genetic data. And you can see here, it's improved a little from 2009 to 2016. 2009, 96% of the data in the Genome-Wide Association catalog was of European ancestry. By 2016, it was 81%, but still overwhelmingly white. It's gotten a little better. This is data from 2018. On the left, we see the ancestry of studies in the GWAS catalog, still majorly European. African ancestry is 9.5%. On the right, if you take it at the level of individuals, it's worse. It's 78% European and 2% African. And so if we're trying to understand the impact of genetic diversity on health outcomes, and we have undersampled African ancestry populations, we are really missing the boat on trying to understand how genetic variation might impact health outcomes, not just for PTSD, for anything. This is just a quick example in the PTSD world. This is a Manhattan plot. That's a genome-wide association study summary, where on the left, in the European ancestry analysis, we got 2 hits on chromosome 6. And on the right, in the African ancestry meta-analysis, we got 1 hit. And the hits were completely different. And if we want to think about understanding disease biology behind complex disorders like PTSD, we're really going to need to get better at making sure that populations other than European ancestry populations are represented. Just to bring it home to be a little bit more current, here's an RNA-seq study on a gene that's intimately involved in allowing SARS-CoV-2 to gain access to your cells. What you can see here is the nasal gene expression of TMPRSS2 is far higher in the sample of African Americans here than in any other population. And it's a small sample set, but again, unless we broadly sample across different diversity, different ancestral populations, including African ancestry populations, we won't understand the impact of biology in concert with social factors that may be shaping risk for disease. So how can we contribute to this? Well, the H3 Africa Consortium is now nearing its 9th year, and the goal of this consortium is to understand the combination of genes and environment in complex diseases within Africa itself. It's been a very productive research effort. I encourage you, if you're interested, to go to the website and spend some time there. We had a symposium last month, and all of the talks are recorded and viewable if you're interested in going into more detail about what those are. Every study that's collecting genomic data is obligated to deposit it into a repository within Africa, and then the actual genetic data that's collected, or epigenomic data in our case, will be deposited into repositories for others to leverage as well. So with this kind of combined effort, we're all working towards addressing this disparity in genomic ancestry information in public databases. So hopefully we can start to chip away at the large disparity that exists between the European representation and all other ancestries. Okay, concluding thoughts. Going back to the very first part of my talk. So we know, at least in the US, there's a higher conditional risk of PTSD among African Americans, and that's true even when controlling for trauma-related characteristics. So we know that other factors, some of which may be social, some of which may be biological, need to be identified that mediate this risk. And some of the ones that have been recently hypothesized, such as perceived discrimination and loneliness, actually did come up in our analyses. But we're really in our infancy when it comes to identifying ancestry relevant factors that prospectively predict traumatic stress. Really, we need to take much bigger strides, but it's expensive, number one, to collect this type of data. And the data has to come from population level samples to really get adequate representation of what's going on. But with efforts like H3 Africa, we are going to make inroads, even if it's slowly, to our understanding of the genetic influences and epigenetic signatures of complex disease. And so with that, I would like to acknowledge my current funding and my many supportive lab members. And I'd like to thank you for your attention, and I am happy to take any questions.
Okay, thank you so much, Dr. Uddin, and we'll have to imagine the 80 people who have been watching who are applauding you, and in particular, thank you so much for the rich and accessible talk. Those are hard things to do at the same time, and I'm not a scientist, and I felt like I followed along, so I'll count that as success. So we do have a few questions in the Q&A box. I'm going to encourage folks who've been listening to the talk to feel welcome to add in questions, but I will start with the two that we have here to sort of get us started. So the question, what is the key difference between W3 and W2 PSS values? This entire study occurred over 4 years, so the time frames between the waves aren't large. Why would W3 be so predictive?
Great question. So the part of the study that we sampled was from waves 1 to waves 3 to predict wave 4, and the interval between each wave was approximately 1 year. And so wave 3 was the wave immediately prior to wave 4, and it was the most predictive. PTS symptom severity in wave 3 was the most predictive of wave 4 PTS symptom severity. In other words, immediately prior psychopathology was most predictive of future psychopathology. Then it went down to wave 2 and then further wave 1. So it's almost like a chronic course, you could say, of PTSS. And that's consistent with the literature. Whether you measure it as PTSD or PTS severity, it is true that prior PTSD tends to kind of portend future PTSD.
Okay. How does the larger W3 value indicate the short-term limit of the epigenetic effects? That is, if W2 has little influence, has its effect faded quickly? And are we seeing the most recent influence of more recent trauma?
So we have epigenetic measures from wave 1 and wave 2. We don't have epigenetic measures from wave 3. That's one of the years that we didn't sample blood with venipuncture. We have blood spots, but those can be difficult to work with. So it is, again, sort of the most immediate wave prior to the outcome wave that seems to be the most impactful in terms of its effect on psychopathology, in this case, PTSS. And you see that by the percentage of contribution it's making to the prediction. You know, the lowest was 88%. So all the predictive value can scale to 100. It's sucking up most of that prediction. And again, we did have prior traumas included in the model, but they weren't ranked nearly as highly. So cumulative trauma was in that set of 95, but it was further down. And so one thing I didn't talk about is, you know, we didn't measure it, but personality could have something to do with how you appraise trauma, and traumatic appraisal may be factoring into continued symptom severity. If the trauma itself isn't ranked highly, still one's perceptions and memories of it may still be carried forward.
So the questioner says, sorry for the very basic question, but is DNA methylation directly heritable? Will the grandchildren of a mother with trauma be more likely to have methylated DNA corresponding to the methylization of their grandmother's DNA?
That's a great question. It's not basic. So in theory, when a zygote, when your germ cells are created, all the methylation tags that were in your parents that create the germ cells are erased and reset. And so when an egg and sperm come together, there's about 100 sites on the genome that aren't stripped away of prior DNA methylation levels, and those are in imprinted genes. And those are often genes that have to do with growth-related factors in the case of genes that are imprinted from one sex or the other. But the vast majority of CPG sites should be fresh and new and ready to be kind of following their cellular fate. And so there are now a handful of studies that seem to suggest that the patterns, even from grandparent to child, might possibly mirror each other. Now, mechanistically, how that happens, it's still really unclear. It may not have to do directly with DNA methylation. There could be non-coding RNAs that exist in the gametes that mediate this. It's still kind of getting worked out in animal models, but great question.
How did you get into this research?
Well, that's a good question. It was a little bit of intellectual orientation, but also taking advantage of opportunities that came my way. So as an undergrad, I was really drawn to human biology. It was a program at Stanford that combined analyses of social and biological factors, which I thought was really fascinating. And that kind of framework was something I carried with me into graduate school. In graduate school, I did molecular anthropology. I did non-human primate work, but my goal was to always work in the public health space. So as a postdoc, I worked in a medical school that was good, but not public healthy enough. So I sort of just sent myself to the University of Michigan and found a job. And at the time, they were launching this Detroit Neighborhood Health Study and didn't know anything about biology. So I showed up at the right time. And the intellectual framework that I had kind of carried with me fits so well for something like PTSD, which was the focus of the study.
Great, a good plug for broad and diverse training to help you be able to make the right contribution at the right time. Do PTSD resistant mothers typically have PTSD resistant children and vice versa?
That's a question I'm not sure there's a direct answer to. I guess it could get at the larger question of if someone's at risk in one generation, does it necessarily mean you're at risk in another generation? And I think the best work that speaks to that is probably Rachel Yehuda's work, who's done work on Holocaust survivors. So she's looked at patterns of PTSD in offspring that either match or don't match parents who were exposed to the Holocaust and had PTSD and didn't. And I don't think that there's a hard and fast answer to that. I know that in her most recent work that looked at methylation of FKBP5, she found that there was a suggestion of resiliency in the offspring, at least in terms of anxiety, in terms of offspring who were the offspring of Holocaust survivors who had things like PTSD. But I think that's something I didn't really have a chance to communicate. These trajectories over generations aren't necessarily negative. You know, if you look at some of the animal work, some studies suggest that the impact into the next generation of exposure to prenatal stress is more inquisitiveness. They'll go out into their, outside of their mazes and check things out more than animals who are not exposed.
I'm remembering from your talk the quote, risk or resilience, right? That there's, that these could go in multiple directions. Are there factors other than personality that you believe merit additional study to answer these questions?
Yes, resilience. So resilience is a huge area of interest in traumatic stress research. And it's not the absence of PTSD following trauma. It's its own thing. And it needs to be measured carefully, and you need to have the right measurements in your study. Our study did not have those measures, so we can't account for that, unfortunately. But twin studies have even shown that measures of resilience are distinct from measures of risk for something like PTSD following trauma. And there's so much we don't know. And I think resilience is gonna, or is already almost one of the next frontiers of where to look at in terms of how trauma actually has an impact or not. I think regardless, trauma has an impact, but whether it has necessarily a negative impact.
From one of our colleagues in the biology department, thank you for the very thought-provoking talk. Can you tell us more about the scientific capacity building taking place as part of H3 Africa?
Sure. So there were 2 rounds of funding. We were funded on the 2nd round, but this consortium has been in existence for almost 10 years. So I will tell you what I am familiar with, but it's definitely something you should check out on the website if you're interested even more, because I'm certainly missing some things. But each study is expected to have some component of scientific capacity building. In our case, we focused on trainees, so recruiting a PhD student and a senior scientist who now is an assistant professor at the London School of Hygiene and Tropical Medicine. He really has outgrown us all. And also equipment acquisition in Rwanda, because ours was a smaller study, so we couldn't really scale up too much. Otherwise, we wouldn't be able to do the research. We were expected to do both. But the program projects that are funded through H3A, which are much larger, I'm talking like $10 million, they have a lot of, in particular through universities in South Africa, bioinformatic scientific capacity building, where they've developed trainings, many of which are online, to get students and postdocs in Africa brought up to speed on the most current methods so that they can then deploy them wherever they are within Africa to analyze genomic data. I mean, wherever you go, whether it's in Africa or elsewhere, the bottleneck for all this work is analysis. And there are very few people that understand both the biology and the analytic components. And H3A is investing heavily in scientific capacity building in that way. And so people outside of Africa often ask to register for some of the courses because they're so well done. And yeah, I think that's one of the major ways.
From another colleague in the biology department, in the Rwanda study, would it be possible to gather or add data from siblings born before the massacre to contrast with those after?
So we did consider that, but then it would be difficult to disentangle their own exposure to the genocide, right? Because they would have lived through it to some extent. Plus, working with a limited budget, so but we did think about that. And in fact, I'm on the committee for a PhD student at a university who is incorporating that aspect into her work, not the biological aspect, but the psychological aspect. And so she's at Montclair State, and she should be able to disentangle some of those questions.
Great. Would you qualify COVID-19 and the pandemic as a broadly traumatic event in that it is life-threatening?
Most definitely, yes. I mean, one type of trauma is a natural disaster. And if COVID-19 is not a slow-moving but deeply impactful natural disaster, I don't know what is. And yes, a lot of that is politically exacerbated, but we have to live with all of the effects nonetheless. And so there's a ton of work right now being done around traumatic reactions to COVID-19, and a lot of work in disparities too. There were many, many calls that came out in April. It was almost like a whirlwind of supplemental funding that you could apply for in relation to COVID-19. So there is going to be, I think, in a year or two's time, a lot more of that coming out.
This is a question for me. In my first year seminar, we read some work by Dorothy Roberts, who was sort of raising some concerns about importing the political category of race into scientific study generally and studying of the genome particularly. And so I'm wondering if you could reflect with us a little bit about what you see as what's gained and what's lost, perhaps, by using these political categories of race, either in your research or sort of in genomic research generally.
Yeah, that's the tension, right? Is race a social construct or not? Well, what we know is that humans originated in Africa, and that is the continent that has had the most time for genetic diversity to emerge. And so if you're looking to capture genetic diversity of humanity, you would be remiss if you didn't look in Africa, because anytime that you identify something as a risk variant in a European ancestry population, well, that might be because the controls in a European population all have one kind of allele, which is a certain type of genetic variant at a particular locus. But if you go into Africa, it's quite possible that maybe there's a variant just like that that raises risk for the same disorder, but you haven't sampled it, right? And I think it is a problem to describe Africans as monolithically Black in the way that that encompasses their genetic diversity, because, for example, in Rwanda, it's not known. There has been no deep sequencing of individuals from that population. The closest there is Uganda. That's been fairly well done. But there are likely a lot of private genetic variants that exist only in Rwanda that we don't know about. So there needs to be much broader sampling of variants across the continent. We can't assume that sampling Ugandans is representative of all East Africa, or sampling, you know, Nigerians is representative of all West Africa even. There's been so much more time for diversity to accumulate that you have to look deeply. And I don't know if that answers your question, but.
It does, thank you. We've got one final comment that I'll read from a local, a person who's working locally here in Walla Walla on resilience. And she said, community resilience work tells us how important the contextual aspect of community building is. So she's glad that you're raising resiliency as a benefit. So I think we are at our time for today, but I really want to offer my appreciation to Professor Uddin for sharing your expertise with us in our panel today. Professor Knight, did you want to jump in?
Yeah, I wanted to echo your thanks there, and thank you for taking the time and for bringing your expertise to us and allowing our students to see public health, biology, psychology, anthropology at work. So yeah, thank you very much.
Thank you. Thanks for the invite. I enjoyed it.
And I would encourage folks to check out the resources that Professor Uddin referenced for additional reading on the topic. Thank you so much.
Great.
Have a good evening.